So, what if you had the complete genetic blueprint for a brand new species? We're talking billions of letters of raw DNA data. What do you even do with that? Well, today we're going to decode that entire process, how we turn that chaotic stream of data into knowledge that can literally change the world. All right, let's get into it. Right, so a team of scientists finishes a sequencing run. They've got the code. But it's not some neat, organized book. Oh no, it's more like they took a phone book for an entire country, put every single page through a shredder, and then just kind of randomly taped it all back together. So the biggest question is, where do you even start? And that's exactly what that first draft of a genome is. It's that shredded blueprint. It's a jumbled up billion-piece puzzle that's probably full of mistakes, gaps, you name it. So our very first job, before we do anything else, is to figure out if the blueprint we've just pieced together is even remotely accurate. To tackle this massive job, it all boils down to two key questions, and you have to ask them in this exact order. First up is quality control. Is the blueprint we've built actually correct? Is it reliable? Then, and only after we're sure about that, can we ask the second question, which is all about annotation. Okay, so what does it all mean? So let's really dig into that first absolutely critical step, because think about it. You would never, ever build a skyscraper from a flawed blueprint, right? Well, you can't build groundbreaking science on a flawed genome. This quality control part, this is where every single big discovery really begins. So, how do scientists actually check the blueprint's quality? Well, they have this amazing digital toolkit. First up, you've got a tool called QUAST. It basically tells us how big our blueprint is and how many pieces it's in. Is it one giant sheet or 1,000 tiny scraps? Then there's BUSCO. Think of BUSCO as the ultimate parts checklist. It checks to see if all the essential, must-have genes that we expect to be there are actually there. And finally, a tool like Mercury is like a high-powered microscope, scanning for tiny little typos letter by letter. And check this out. This is a perfect example of a great report card from BUSCO. See that 96% complete? That tells us that almost all of the absolutely essential genes we expected to find are present and accounted for, fully intact. Getting a score like this is a huge confidence boost. It means our blueprint isn't missing any of its most critical pages. In fact, that number, greater than 95%, that's kind of the gold standard. In the scientific community, if your BUSCO score is over 95%, that's the green light. That's the signal that says, okay, this blueprint is solid. It's reliable. We can build on this. It's really the benchmark that separates a rough draft from a genome that's ready for real discovery. And really, this quote says it all. The bottom line is everything that comes next, finding the genes that cause disease, figuring out how evolution works, all of it, every last bit, rests on the quality of this first step. You have to have a strong foundation. Okay, so we've got a blueprint we can finally trust. It's accurate, it's solid. Now the real fun begins. It's time to figure out what it all means. This next phase is called Genome Annotation, and it's basically a massive treasure hunt for the actual instructions, the genes, that are hidden somewhere in those billions of letters. And it's a really methodical process. First, you use a tool to find all the repetitive bits of DNA. You can think of this like all the boilerplate legal text in a contract. It's just noise. So the next step is to mask all that noise, to just hide it, so we can focus on what's important. And once all that clutter is gone, these powerful prediction tools can scan the clean blueprint and spot the unique patterns that scream, hey, a gene is here. And after all that work, boom, what you get is a detailed map, a list of coordinates pointing to where every single potential gene is located on your blueprint. But of course, that immediately leads to the next big question, right? I mean, knowing where a gene is doesn't tell you what it's for. We've found the address, but what does the building do? What's its purpose? Does this gene build a protein for seeing color? Does that one help fight off infections? This next step is all about assigning a job to every instruction we found. And this is what we call functional annotation. We're literally going through our blueprint with a label maker. We're pointing to different sections and saying, okay, this part right here, that's the engine. This whole section over here, that's the transmission. And this little bit, that controls the brakes. We're systematically adding meaning to the map. So how in the world do we know what labels to use? Well, we use these incredible digital detective tools. Things like BLAST or Diamond take each one of our new mysterious genes and compare it against a massive global library of every known gene from every other organism. So if our gene is a 99% match to a known engine gene from a fruit fly, well, we can be pretty confident that we've got an engine gene too. Other tools look even deeper, identifying smaller functional parts, kind of like the specific pistons and spark plugs inside that engine. All right, let's just take a second and recap. We've gone from a shredded, chaotic, billion-piece puzzle to a fully assembled, quality-checked, and clearly labeled blueprint. It's a huge amount of work. But why does it matter so much? Why go through all this trouble? Because this is why. This is where the magic happens. A high-quality, annotated genome is what allows us to develop personalized medicines tailored to your specific DNA. It's how we can engineer crops that can survive a drought. It's how we trace the genetic roots of diseases like cancer and Alzheimer's. And it's how we uncover the deepest secrets of how life on Earth evolved. This foundational work makes all of that possible. When you step back and look at the whole journey, it's just incredible. We started with complete chaos, a puzzle with a billion pieces. And through this meticulous process, we've turned it into an organized, trustworthy guide, a reliable map that scientists can now use to explore a whole new biological world. What this process really means is that we've learned how to read the book of life for any organism on the planet, and that gives us a power we've never had before in human history. So it leaves us with a pretty profound question. Now that we can read the story, what new chapters should we write next?